Hi students, today we will discuss for the process that is called as transcription. It's a process in which the copying of genetic information from one strand of the DNA into RNA it takes place. It is the first step in gene expression, and here the RNA it is formed from DNA. And the enzyme which is responsible for the process that is called as RNA polymerase, in which the nucleotides they are joined together and the RNA strand it is formed. RNA polymerase polymerizes and the linking of the nucleotides leads to the formation of RNA strand. Now, in this process which is called as transcription, here only one strand of DNA it is involved. In contrast to the process that is called as replication, if you can recall, in the replication both the strands of the DNA they are involved, but in the transcription only one strand of DNA it is involved. And the reason behind that, if both the strands of the DNA they are involved in transcription, then what happened? Two mRNA molecules will be there, resulted in the two polypeptide chains, and you can say here that from one DNA molecule two proteins will be formed, and the formation of the two proteins from single DNA molecule it will hamper the genetic information transfer machinery, right? Genetic information transfer machinery will be disturbed. So this is the reason the two strands of the DNA they are not involved. The second reason, the first reason we have seen, the second reason is what? The second reason is that if the two strands of the DNA will be there, then two RNA molecules will be formed, and being complementary in nature, double stranded RNA would be formed, and this will prevent the RNA from being translated. So, in this case, the exercise of the transcription would become a futile one. So, this is the reason the two strands of DNA they are not involved in the process that is called as transcription. Now, let us discuss for the transcription unit. Here in this diagram, you will be able to see the two strands. One is template strand and the another one is coding strand. What is template strand? The strand whose polarity is 3-5 direction, that is called as template strand, right? And the another one where the polarity is 5-3 that is called as coding strand. So this one is template and this one is coding strand. Polarity matters. There is a convention in defining the two strands. And one more point you should be very clear with, that the DNA dependent RNA polymerase, it catalyzes the polymerization in only one direction, and that is 5-3 direction, right? So the polymerase works in this direction only, and these are the two strands. Now, the promoter and the terminator regions, you will be able to see here what the promoter and the terminator regions, they are having their significance. Actually, the promoter and terminator regions, they flank the structural gene in the transcription unit, right? And the polymerase enzyme, it binds to the promoter region, the reaction initiates, and it is initiated from here and terminate at the terminator region. So these are the sequences actually. Now we have discussed for the polarity of template strand and the coding strand. You know, it is the position of the promoter region which determines the strands. What I am trying to say here, that promoter region, you know, it is located towards the 5' end of the coding strand. Always remember, it is located towards the 5' end of coding strand. And the terminator, it is located at the 3' end of the coding strand. And if the promoter and terminator switches their position, so the strands, they are also changed, right? So it is the presence of a promoter in a transcription unit that defines the template and coding strand, and the promoter and the terminator, which are the sequences, they flank the structural gene here. This is the transcription site. There are some other sequences are also there, which are called as regulatory sequences. Regulatory sequences, they are present upstream or downstream to the promoter, and they regulates the process. Now, the template strand, it is also called as antisense strand, non-coding strand, minus strand, and the coding strand, it is also called as plus strand, sense strand, and non-template strand. Coding strand, interestingly, don't code for anything, and it is just equal to RNA. Only the change is for the presence of uracil. In the strand, coding strand, the thymine it is present in place of uracil, which is present in the RNA. Now, come to our discussion for the gene. You know, from very beginning, we will be able to see that genes, they are the unit of heredity. They are considered as the unit of heredity, and they are responsible for the transmission of characters from one generation to another. That is why it is called as the functional unit of inheritance, which is the process. Now, what is gene? And the DNA segment which codes for the protein or tRNA or rRNA, they are considered as genes. All in all, coding DNA sequences, they are called as genes. But if we talk about that how the gene control the characters, that also we know gene, they codes for protein, and that is how the controlling of the character takes place. So the DNA segments which code for the protein, they are called as cistron, right? In general, the coding DNA segment, it is called as genes. It can be for tRNA, it can be for rRNA, but particularly the DNA segment which code for protein, that is called as cistron. And in the eukaryotes, monocistronic condition is there, and in the prokaryotes, polycistronic condition is there. In the monocistronic condition, the split gene arrangement is there. What the split gene arrangement is? Where the exons, they are the coding sequences which are expressed on the processed RNA. These are the coding sequences. They are intervened by the introns. These are the non-coding sequences which are not expressed on RNA, right? So this is what the split gene arrangement, and the split gene arrangement, it complicates the definition of a gene definitely, right? And it is present in the eukaryotes because in the processed RNA only exons, they can be seen. Introns, actually they are removed, and the regulatory sequences which are there for the regulation of the process, either upstream or downstream to the promoter, they are also responsible, like they are also having the significant role in the inheritance of a character. And the regulatory sequences, sometimes they are defined as the regulatory genes as well, despite of the fact that these sequences, they don't code for anything. They don't, like these sequences do not code for any RNA or protein. So gene, it is used as a general term for the DNA sequences. Sometimes for the non-coding sequences also, the genes term, it is used. So we are done with the first topic here, like what the transcription is and what the transcription unit it is made up of, and how we can define a gene. What actually genes are? Genes, they are nothing but the DNA segments. Now we will be able to continue our discussion in the upcoming videos for the same topic. And till then, have a very good bye.